So good evening everyone, and welcome to the latest in our inaugural lecture series here at Keele, which are always a highlight of our academic year. My name's Christian Mallon, and I've got the privilege of being the Executive Dean for the Faculty of Medicine and Health Sciences. Professor Melissa Bowerman is our Professor in Neuromuscular and Skeletal Disorders in the School of Medicine and a group member of the Wolfson Centre for Inherited Neuromuscular Disease at the Robert Jones and Agnes Hunt Hospital in Oswestry, which is one of our key academic partners for both research and education. Melissa completed a PhD at the University of Ottawa, where she held a prestigious Frederick Banting and Charles Best Doctoral Training Fellowship, in addition to a Ronald G. Wharton Researcher in Training Award. So two very, very prestigious and highly thought of training fellowships. She then worked in Montpellier before moving to Oxford to work with Professor Matthew Wood's group. Her research programme aims to identify novel pathological pathways that contribute to the pathophysiology of rare genetic neuromuscular and skeletal disorders, and to investigate novel therapeutic approaches using a combination of omics, bioinformatic drug repurposing strategies, and dietary interventions. This research is absolutely critical. Rare diseases are only rare until you have one, and then they become the focus of your life. Research in rare neuromuscular and skeletal conditions is neglected, and Melissa's work is giving hope to people with conditions that many people have never even heard of. Melissa's also committed to developing the next generation of clinical academic researchers. Her leadership in the medical school of the Aspire and Intercalation programmes is a really critical part of this, providing hands-on opportunities for clinical students to get involved and inspired by research. Melissa is a true leader with an international reputation for her research excellence. She's also a dream to work with, always going the extra mile to support colleagues and students. I've never once seen her without a smile on her face. Even tonight, Melissa is smiling. Even when I jabber on to her about ice hockey, which apparently is hockey, not ice hockey, and mispronounce my favourite treat as poutine. It's a great privilege to invite Melissa to deliver her inaugural lecture. Melissa.
Thank you, Christian, for the kind words, and thank you, everyone, for being here. I'm very touched by seeing you all being here for me tonight. So, as Christian mentioned briefly, this has been my journey. I've put us so far. I've had no intention of leaving. However, life has taught me that you never know what will happen. But we plan to stay. And so for the next 35 to 40 minutes, I'll just walk you through our journey that started in Ottawa. So I was born in Ottawa, Canada, and I also did my PhD there at the University of Ottawa / Ottawa Hospital Research Institute. And I joined Rashmi Kothari's lab as a master's student. So in Ottawa, well, same as here, you join as a master's student and you can transfer halfway through to a PhD. And at that point, I didn't really know where I was going to go, but I knew I really liked biology, I liked research, I liked science, so I applied to different PhD or postgraduate positions that were offered at Ottawa and got an interview with Rashmi. Went to his office, really didn't dress up for it. I was ill-prepared, but just sat down. He talked about the different projects in his lab, and I was hooked, and I chose one, and he gave me the position. And that project that I got hooked on over 20 years ago was on a condition called spinal muscular atrophy. And that condition is a rare condition where in the spinal cord you lose the motor neurons that branch out and connect to your skeletal muscle and allow them to function. So those motor neurons, they die in spinal muscular atrophy, and that leads to severe muscle loss and in most severe cases paralysis and early childhood death. It's a disease that's caused by the loss of one gene, and I bring it up here because it'll be important in a few slides, but the loss of the survival motor neuron gene causes SMA. It's an autosomal recessive disease, so each parent has to give one of the defective copies for SMA to occur. It occurs in 1 in 6,000 to 10,000 live births, and that's why it's defined as rare. But interestingly, the carrier frequency is quite high. So 1 in 40 individuals carry that defective allele, but having one is not enough for you to demonstrate disease. So I've been working on this condition for over 22 years, hence why a little bit of background. Now, not a lot of people are familiar with SMA, but hopefully in the UK, some of you have been given awareness because Jessie Nelson, who was a singer in Little Mix, recently announced that her 2 twin daughters were diagnosed with SMA. So that's raised awareness, particularly here in the UK. But because it's a childhood condition and because previously without treatment, children would die early, not a lot of people are aware of it. But Jessie Nelson is now an ambassador for SMA UK and is doing a great job at helping us raise awareness. There's different clinical manifestation of the conditions, but it's a childhood condition. So in the most severe cases, the children die before the age of 2, but now there are treatments, but 60% of the cases are the most severe case population. Then there are type 2 and type 3 patients that have a variety of different neuromuscular impairments depending on the severity. And really, I won't go into the details here, but please Google these treatments, watch a YouTube a video, because in 2026, we're in this wonderful state in the SMA field where there are 3 therapies that are available for people with SMA. And the goal of these 3 therapies is to increase SMN, so reintroduce SMN that's lost. And they're life-changing and they're life-saving therapies. So it's a great time to be in the SMA field. But I'm going to leave it at that because when I started, there was none of this. And so we were still asking the basic questions of what's causing the pathology and how can we treat it. So one of my questions when I started in Rashmi's lab was really to understand why SMN, why loss of it caused that death of the motor neurons. We still didn't know in 2004, 2006 why. So a lot of research groups were interested in that. And that was part of my work. And a lot of experiments further brought us to the identification of a particular pathway, so specific proteins that were dysregulated, that were impaired in SMA neurons. And you don't need to focus too much on them, but they were called ROA-ROC. But importantly, their role, and you don't need to focus on all of this, but their main role is to provide cellular stability. So they give that cell stability and function, and when that pathway is impaired, you lose that function and stability. But what was most important from that work is there were 2 actually drugs that were available that target that pathway. So one of them was the Y compound, Y27632. That's just a chemical compound that you can purchase. And Fastizil is actually a drug that's currently used to treat stroke, and that acts on that pathway as well. So we wanted to ask, if we target that pathway in animals that have SMA that we work in the lab, what does that do? And so I've summarized 2 papers in a few figures, but just to give you the take-home message is that when we treated animals, so in the lab we work with mice that have SMA symptoms, and that allows us to understand pathology and effective treatments. And when we treated these mice with Fastizil, so all the straight lines above are animals that do not die. In green are SMA untreated animals, so every step down is a death of an animal. And in blue is a treated animal. So we were able to improve survival of these animals, which was really big at that time. Then we sent it out for a review, and then one of the reviewers said, what's the effect of this drug on muscle? And at that time, I complained a lot because we were just focused on motor neurons. That was, in SMA, we thought only the motor neuron was impacted. So I was a bit grumpy saying they've asked us for a stupid experiment. We're just going to do something for nothing. But, you know, a spoiler alert, it was a really, really good question. So what we did is we looked at muscle size. So this is a cross-section of skeletal muscle. In purple are all the different muscle cells, and we basically quantify the area of that muscle cell. So these are non-SMA animals. These are SMA animals, so hopefully you can see that the purple circles are much smaller. And actually, when we treated with Fastizil, we totally reversed the muscle loss that occurs in those mice. And that was quite a surprise to us, but also the community at large. And these were part of the first studies that demonstrated drug repurposing, so the idea of taking drugs that are used for other conditions and using them in your own condition, and non-SMN targets. So back then, the idea was to design drugs that would upregulate SMN, which makes complete sense, but this was a hard sell, this study of drug repurposing and targeting something out of SMN. Now fast forward to 2026 and it's a hot topic, okay? So you go to conferences, you have whole sections on that, you have reviews. This is where the field is at now that we have those 3 gene therapies that upregulate SMN. But we were the first to demonstrate the potential of this. I had a very long PhD for many reasons, including the birth of my kids, and in Canada, it's longer than 3 years. So I found my way from starting the motor neuron question and then made my way to the pancreas. And a lot of people, including myself, might ask, how did you do that? Well, it was an accumulation of anecdotal evidence throughout several years, whereby we were trying to make this new mouse model that had SMA symptoms. And by doing so, we had a range of animals that had a little bit less SMN than in control animals, but not enough to have SMA, but they had reduced levels of SMN, and we aged them for a variety of reasons. And these mice, as they got older, they started to have these metabolic symptoms. So they got fat, their urine, their pee smelled, and those are usually kind of signs of diabetes or metabolic issues. But at the time we ignored that. We said, we're not interested in that, we're just interested in the neuron. Then we had collaborators in Ottawa that took the pancreas from our mice and analyzed the islets. So the islets in the pancreas, they regulate your glucose, so bring them down when it's too high and vice versa. And they found defects. And again, we kind of ignored that, thought it wasn't important for SMA. And then finally, we treated animals with resveratrol. Resveratrol is a natural compound that acts on metabolism. And when we treated our animals with resveratrol, it made them worse. And so there was this metabolic kind of issue that kept coming up. And I remember Rashmi being away for 2 weeks and printing a lot of papers and reading and building my case so he would let me look at the pancreas and that glucose. He came back, I went to his office. I think I talked for 2 minutes and then he said, okay, just do it. So off I went on this exploration of pancreas and glucose and SMA. And I'm gonna cut this story very short, but we ended up publishing for the first time glucose and pancreatic impairments in SMA mice, but also in tissues from people with SMA. And the 2 key important findings from that study was the impaired glucose metabolism. So you might have done that in a clinic, taken a bolus of glucose to see if you can get rid of the glucose in your blood. So we can do that with animals as well. So in wild-type blue animals, you give a bolus of glucose and the glucose levels spike up. But then the wild-type, the control animal, is able to clear that really quickly from the blood, where the SMA animals took longer to clear that. So there was a glucose metabolism defect. And then we went back to the pancreas islets. So these are the functional units of the pancreas. So you have in red are the insulin-producing beta cells, and in green are your glucagon-producing alpha cells that help you regulate glucose levels in the blood. So these are all islets from the wild-type animals. And then when we looked at the SMA animals, hopefully you can see that the normal pattern of red in the center and green outside was completely disrupted in the SMA animals. So the islets of the SMA animals mostly have the green glucagon-producing cells. And again, we were the first to kind of show the metabolic and non-motor neuron issues in SMA, because again, everyone was focused on the motor neuron and the muscle. And now you can Google SMA glucose metabolism defects, and there are several case reports and case studies in people with SMA. And also, we all now understand that SMA is beyond the motor neuron. So if you look at any cells or tissues in the body, it's also impacted to varying degree. And that makes sense because SMN, which is lost in SMA, is found in all cells and tissues in the body. So this is also a topic that's not disputed anymore. But back then, it was really difficult to push that idea of glucose metabolic issues. What the most important part that came out from this study, though, is a long-term friendship and collaboration with Mary. Mary, when we published the glucose and pancreas study, called me. She's the mom of Krista, that's right here. Krista has SMA. And Mary was adamant about metabolic issues in SMA all her life, and develop a specific diet for her daughter that is now embraced by a lot of families. But the clinical teams weren't listening to her, so she emailed me. We started to chat. We've never stopped chatting since then. We talk about every 2 to 3 months about SMA and metabolism. And she got us connected with one of her good friends, Anne McGuire. So Anne McGuire is the president of SMA Angels Charity, and she has 2 granddaughters, twin granddaughters that have SMA. One, unfortunately, sadly passed away when she was young, but Skylar will be 30 this year. So it's really been a 20-year-old friendship and relationship with Mary and Anne. But the most important highlights from Ottawa, and I think I've alluded to them previously, is the birth of my kids. You might have spotted them here today. I won't embarrass them, but Olivia was born in 2008 and Alex in 2010. And I took two 6-month maternity leaves during that period. Okay, so Ottawa was great. It was where we were from. And then we ended up 2 years for my first postdoc in Montpellier, and it is as nice as this. And so how did we get there? Well, I wanted to do a postdoc internationally, and I said to Jeff, my partner, because he had been working in the government for 17 years, and I think he thought we're going to live for the rest of our life in Ottawa. So I said, if we were to move somewhere for a 1-2 year postdoc, where would it be? And he said, south of France. And therefore, I sent CVs south of France. So from Nice to Marseille, I sent CVs. I arranged interviews, and I got on the plane all by myself and traveled from Nice to Montpellier to do several interviews, and really clicked with Cédric Raoul. Cédric actually was in Marseille, but he moved his lab to Montpellier, which I was happy about. I liked the project he was offering, he liked me, but there was no funding. So we started to apply for postdoc fellowship funding. And one of them was the long-term, the EMBO long-term fellowship that we applied for. Now the format might have changed now, but back then if you got through interview, you got set up with one of the members and they called you for an interview. And you know who's going to call you. So I knew that Professor Dame Kay Davies would call me. For those of you that are not in the neuromuscular world, she dominates that world. She was editor of Human Molecular Genetics. She's a superstar. It was very scary. Rashmi told me she would be scary, so that scared me even more. But she called, we had a chat. She did ask me, why are you going to Cédric's lab, which was a good question because he was just starting himself, and the purpose of the question was really about why not go to a more established lab. I could not say, well, because my partner only wants to go to the south of France. But I did end up managing an okay answer because I was awarded the EMBO long-term fellowship that allowed me to go to Cédric's lab. That means I stepped away from the SMA field for 2 years. I went into the ALS motor neuron disease field, which in some instances is similar to SMA because you've got the lower motor neuron component, but also you have the upper motor neuron component that is lost. So the upper motor neurons are lost in ALS, but you have muscle wasting, muscle loss. It's an adult onset disease, and because of that, more people are aware of ALS motor neuron disease. So particularly here in the UK, we've got the rugby players, Audie Weir and Rob Burrow, Stephen Hawking, and hopefully most of you are aware that Eric Dane, who played in Grey's Anatomy, recently passed of ALS. And so that raises awareness and the knowledge of what ALS and motor neuron disease is. Again, don't worry too much about this side, but basically Cedric's plan was for me to look at this pathway, tweak FN14 and ALS. And Cedric really liked the spinal cord, so he wanted me to focus on that pathway in the spinal cord. But as I'm doing my reading, because I like to do reading on the side and build new ideas, I find that actually this pathway is also important in skeletal muscle, and I had learned my lesson in SMA that skeletal muscle is important. And so I asked Cedric, can we look at muscle as well? And he was adamant, no. So I did things a little bit on the side and in the secret and built my project. And at one lab meeting, I went up to him, I said, well, this is what I've done. What do you think? And he was very happy because we published on this pathway in both the spinal cord in the muscle. So I was happy that I kind of stuck with my guns and kept looking at this pathway. So what are the most important highlights from France? Well, France. Those of you that know me personally know that I talk a lot about France. When we first moved as North Americans, we wanted to travel all of Europe, and then we realized you just drive one way for an hour in France and you're in a different world, and it's wonderful. So we explore numerous areas in France. In our second year, we bought a house. Those of you that know me, I talk a lot about our house in the south of France in this little village called Soumont at the end of a road. This was at the beginning when Jeff tore all of the floors down and rebuilt everything. It had no electricity or plumbing. I think we just got plumbing in a few years ago, but it's been a project. We lived in wonderful places, so by wineries. We got chickens for the first time. That was such an interesting and lovely thing to have. We even moved the chicken coop that we had painted. And when you live in the south of France, everyone visits you. Everyone visits you. So you'll see we're sitting at this table a lot, but we had families and friends that would visit us quite often, and it was quite a pleasure to welcome them and discover this part of the world together. We were able to reunite with some friends that lived in France that we had met in Europe, and we made new friendships through work and through the kids' school that we still maintain and see every time we go in the summer. Okay, and then we had to leave south of France. Why, you might ask? Well, the funds ran out in the south of France. So my EMBO fellowship was only 2 years, and Cedric unfortunately didn't have enough funds to keep me. So I was offered a position in Montreal, which is 2 hours from Ottawa, our hometown. But there was this house in the south of France that we really, really love, and that flight between Montreal and that place was a bit too far for us to realistically think that we would go back. So I applied for a position at the University of Oxford in Matthew Wood's lab. And interestingly enough, at the interview table was also Susan Hammond. Susan Hammond did a PhD at the same time as me in an SMA lab in Chicago, so we saw each other quite often. So it was a great pleasure to be interviewed by her, work with her, and we're still very, very good friends. So life does that sometimes. In Oxford is really where I developed my independence. So I was hired on a project by Matthew Wood to look at cell-penetrating peptides. So there's small peptide sequences, you attach a cargo to it, and it allows you to bring that therapeutic cargo to the cell. So I was hired to do that, but that's not what I wanted to do with the rest of my career. And because it's an Oxford lab and Matthew had a lot of money, I had a lot of flexibility to actually develop my independent career. And he was not interested in what I was doing at all in that sense, so I had full liberty to develop that. And I was interested in both SMA and ALS, understanding what cells and tissues outside of the neuromuscular system, the neurons and the muscles, were impacted. I was interested in the metabolic pathways, glucose, fatty acid, amino acid, lipids, how they're impacted in SMA. We dabbled in circadian metabolism, so that's how your 24-hour cycle is regulated by night and day. But really is once we understand how all of these different metabolic issues and tissues are impaired in SMA and ALS, is can we target them using either drug repurposing, drugs that are already used in other conditions, exercise, or even diet. So use approaches that are more accessible and less expensive than some of the newer therapies. So I was able to develop those ideas, but also to apply for money as an independent researchers. For the most part, there were small pots of money, so they were between 10 to 50,000, but they were under my name as PI, building that kind of capital and evidence that I was able to get that funding. It all started with SMA Angels. So Anne first gave me a donation, and she still gives me donations to this day that help me build my foundational work, and then applying to these different patient organizations. What are the most important highlights from Oxford? I got to meet Professor Dame Kay Davies. She's in Oxford. I got to attend one of her lectures where, she shared that, when she was first offered her job, that she had to ask her husband if she could get the job. But I also got the opportunity to go to a women's dinner with her, sit beside her, and remind her that she had interviewed me for the EMBO Long-Term Fellowship. So that was a very special full circle moment for me. In Oxford, I received a junior fellowship at Somerville College, which means that I could go to lunch at the high table every day. And for a North American to be at that kind of Harry Potter style table every lunch and go to evenings was very, very special as well. My kids started to play hockey. Yeah, I know you say ice hockey here, but it's only hockey in Canada. And this is important because they still play. And I think I've seen, and we've seen all of the rinks in the UK, and they're pretty far apart. People come and see you, but less in England than they did in south of France. But the hardcore people still came to visit us. We got to reunite with my good friend, Lindsay Murray, who was a postdoc in Rashmi's lab when I was finishing my PhD, and she's now in Edinburgh, so we see each other often, but it was great to reconnect with her and made new friendships as well, some of which that we still maintain and I visit regularly. And then I got here at Keele University at the School of Medicine. How did we get there? Jeff said, get a job. That was enough. That was enough of these permanent, unstable positions. So he says, it's time for stability, get a job. So I applied at multiple places in Canada at Keele, not knowing where Keele was until I had applied and kind of located on the map, and applied at the School of Medicine and got selected for interview. Interestingly enough, the post was for someone with an expertise in endocrinology, which I do not have. But I had just attended a lot of these sessions where we were told that as women, we're less likely to apply for jobs unless we meet all the criteria, but men do. So that was in my head. So I'm like, I'm going to still apply. And I got interviewed and I got the job. And I do think Gordon Dent was at that table and he did ask me about, are you able to teach endocrinology? And I think it was a good enough answer. But I think it was also making them realize that, maybe you're the person that they didn't know they wanted, really. And on the train back, I got offered the job here. And I've never regretted it. So since then, I've been developing and establishing my research program at Keele, which is really aimed, as Christian said, at understanding the pathological mechanisms in rare diseases, but also how can we go ahead and treat them with different approaches, focusing primarily in the last years on ALS and SMA. What I just want to show in the next few slides is kind of one of the studies I'm most proud of is this. We always talk about bench to bedside, doing something in the lab and then bringing it to the patients. And this is one example that we looked at. So we looked at dietary, they're BCAAs, branched-chain amino acids, so leucine, valine, isoleucine. They're in your meats, your dairy products, but you can also buy big powders of them because weight builders will use them to build muscle. It all started again with Mary and Anne, because part of that diet that Mary established was an amino acid-based diet. And so we first worked with them, but also with our animal models to understand, why is this diet working? What's happening in SMA tissues and cells that explains why this diet is working? And we had a good idea of why that was. And when we gave that diet to our animals that have SMA, it improved a lot of their phenotypes. And so we searched a long time for a clinical team that would work with us. So we worked with Giovanni Barinello's team in Gosh, and Graham O'Connor, who's the dietitian. And interestingly, the dietitian is not in the SMA field, so he didn't think the diet would work. He was anti-diet, which was quite nice because he was very unbiased to the study. But we set up a study with 12 individuals with type 1 SMA, the most severe form, but they had all received one of those SMN treatments. So they were all on one of those SMN enhancing treatments, but they still have some symptoms. And the plan was to put them 8 weeks on an amino acid-based diet, and then the next week put them on a whole protein diet, which is completely different. And one of the key results is after 8 weeks, none of the patients wanted to go back to the whole protein diet because they felt much better. And part of the reason that they felt much better is most of these patients were still on GI type of medication because they had a lot of digestion issues. And actually just changing the diet improved their digestion so much that they were able to not take the medication anymore. And that's really big actually for quality of life in individuals with SMA. So we were really happy that we could bring to light and to the bedside our BCA diet. We've also been building long-term partnerships with charitable organizations. Most of our funding comes from patient charities. We work with them and go to public events and talk about our research quite frequently. And without them, we would have never been able to do, none of our research. We're always very grateful to them for supporting us. And it's allowed us to go in this new direction. These are just a few more slides, I promise. And it's acrodyssystosis type 2. It's okay if you've never heard about it. There are less than 80 individuals in the whole world with this condition. It's a rare developmental skeletal dysplasia, which means reduced bone growth. But there's a range of symptoms, including cognitive impairments and hormonal resistance. And we got in touch, or people from Acrodosis Support and Research, so family members got in touch with us because there are actually drugs out there that target the defective protein in acrodyssystosis type 2, but industry wouldn't work with them because there were no models to test the drugs. So we worked with them to do what we do best, is to develop a mouse model for the condition so we can better understand what's happening in that condition and what the effect of the drugs are. So we got funding from GEM to create the mouse model, and since then we've been building that research program and got funding from MRC and GOSH to further develop that. So it's been really great to work with Acrodyssystosis Support and Research directly to build that research program and use our expertise that we've built in the SMA and ALS field. Okay, last few slides are bringing you back to my title, which are kind of like 3 ingredients that I think were important for my research career. One is serendipity. I think I could give you a million examples, but for me, the one that I always stick on is the lab that I ended up in. Particularly in your PhD, that's often unplanned. And so I could have worked on something else, but I worked on SMA and I'm still working on SMA. But also, you don't know if that supervisor is going to be a good supervisor, a positive supervisor, and I was lucky in all of those ways with Rashmi. And I hate using luck because it shouldn't be luck. All supervisors should be supportive and embrace all the trainees, but Rashmi just did it naturally. And that was particularly important for me having my children. Never did Rashmi make me feel less of a scientist for choosing to have kids. Never did he reduce my opportunities. And never did I feel that I couldn't succeed because I chose to have kids. And for some of you, it might seem like it doesn't happen anymore, but these are graphs from a recent paper published in March 2026, where in blue are the line of progression for the men and in red the women. And it's basically progression in terms of research and employment at the university after the birth of a child. So that significantly impacts you. I always say when you apply to funders and they say, did you have a break in your research? And I put my maternity leaves, but I also say, but my kids are still here. I still have to take care of them on a daily basis, and I think that has a lot of weight. A recent study also published, 10 simple rules for postdoc moms to stay competitive in academia. Although I think this was well intended, I don't think it's the role of the postdoc moms to make sure that we stay competitive. We've got to change the systems and environments that support women. So I'm forever grateful, not just to Rashmi, but the two other male supervisors that I had that always supported me. And I think mentorship is really important, if I've not convinced you, because when I was in Rashmi's lab and I started to also supervise and mentor, I always took what I liked of his mentorship and tried to apply it to mine. And that happened with Emma Sutton. Emma Sutton was one of my first PhD students, and I tried to remember what did Rashmi do that I really like, and try to reproduce that with a little bit of Melissa sprinkle, obviously. And when Emma was ready to find a postdoc, Rashmi was looking for a postdoc. So Emma is now in Rashmi's lab doing her postdoc, and when I ran into her mom after a few months, I said, how's Emma doing? And she said, Emma loves it. She said, Rashmi's just like you. And I thought that was the best compliment because I was trying to be like him, but he's like me, so that's perfect. And actually just last month, Emma was awarded funding from Cure SMA. So I really do think that obviously her success is driven by her, but it didn't start with me. It started with Rashmi planting those seeds to success. So be good supervisors and mentors. Risk-taking. It was a lot of risk to move the number of times that we did. We had 2 kids, 4 and 2, when we first moved. We didn't really ask you if you wanted to move. We kind of packed your bags and left. But every move was a months-long discussion between Jeff and I. Are we doing it? All the visa papers, those of you that have moved, everything that comes into it. But also project-wise, hopefully I've kind of given examples of a lot of my projects and research was going the way that we were told not to go towards. There was an easy way to go towards the motor neurons, to go towards SMN. We went towards the pancreas, the muscle, drug repurposing when it wasn't a hot topic. But in fact, I think it's okay to go the risky side when your data are telling you that it's okay. And that's why we do science, because the data tells you if it's okay to go that way. And I'm glad that I took those risks because it's allowed me to build this kind of niche expertise and recognition in the fields. And finally, the power of patient voices. It started with Mary and Anne calling me and telling me that my research was important, and that's really when I started to really talk frequently with patients and families to understand what was important to them and if we could design our research to answer those questions. And now it's grown to this really beautiful community of parents or individuals living with these conditions that I communicate with regularly. Sometimes it's not about the grant that we're putting in, but just having a chat about how they're doing, what's important to them, and building those foundations. So I'm really grateful to all of them for giving their time to me. So hopefully I've shown you, it wasn't this messy, my journey, but I don't like people to think that I came from Ottawa to here knowing exactly what I was doing. Every step was where next, and life kind of decided for us, but it worked well, so we took another chance. And that's what brought us here to Keele. I will end with my heartfelt thanks. I started writing everyone's name and I thought this is going to be endless, and also I didn't want to forget one person's name and then they would point me out. So it's colleagues past and present, collaborators, friends, family, people living with rare diseases and their families, Bowerman lab members present and past, our funders as well. Also my mom, this is a picture of my mom. She passed away 13 years ago. But when we decided to move to France, she was sick. She passed away from metastatic cancer. And I met with her after one of her treatments, and I told her, we're thinking of going to France. Is it okay? Because she was the only person besides Jeff that could say no, and I would have not gone. And she said, yeah, go. And I'm forever grateful for that gift, because that's what you want from any parent, that gift of freedom and go be yourself. And we wouldn't be here had she said, yeah, go ahead, Melissa. So forever grateful for that. And last but not least, these guys, none of it would have been possible without you, or it wouldn't have not been fun. This was when we first moved, because I told them I don't think I could have found a more recent picture that they would have all been happy with now that they're older. So I went for 2012 thinking everyone will be A-okay with that. So with that, thank you for your attention. Thank you for being here.